Hi, this is the recording to complete the slides for the first of two lectures on the cellular level of organization of the human body. Let's get started. In the second slide, what you can see here is the overview of this lecture. So we're going to start with just the basics of the cell, moving on to the composition of the cell membrane and what's on it, and then thirdly to fluid in the body, the different compartments that exist, and we're going to explore the concept of a semi-permeable membrane and tonicity. We're also going to talk about how things move between compartments, and so we're going to look at two different types of transport. We're going to look at different organelles, baby organs that exist in the cell, and then we're going to finish off with a discussion on the cytoskeleton, right? So when you look at the word cytoskeleton, cyto refers to cell, and then skeleton is the framework of our body. So you've got a skeleton, we've all got a skeleton. The cell also has a skeleton, and the skeleton of the cell, it's called the cytoskeleton. So let's start with the basics of the cell. When we look at a stereotypical cell, what we can see is that it can be divided into three main components. So the first component is the plasma membrane. So the plasma membrane, or the cell membrane, used interchangeably, separates out the cell, what's inside the cell over here, from what's on the outside of the cell. So it basically separates out the insides of the cell and what's outside. Inside the cell, what we have is we have a cytoplasm over here, which is a thick gel-like substance inside the cell that contains water. And you can see that inside the cell, there are lots of different things called organelles inside the cell. So organelles are called organelles because they're like little organs that exist inside the cell. And then right in the middle of the cell, you can see here, there is a nucleus over here. And so we'll talk about the composition of the nucleus as we progress on, not only in this lecture, but in the next lecture as well. So the first thing I'm going to talk about is the cell membrane. We've talked about the cell membrane before. We know that it's composed largely of phospholipids. And what happens is the structure of the cell membrane is that it's organized into, it's comprised of two layers of phospholipids that face each other. And that two layers is called a phospholipid bilayer. And so if you look at the structure depicted in the first diagram at the top over here, what you can see here is you've got layer 1 and layer 2 over here. And what you can see here is that with the phospholipid, we know from last lecture that it's composed of a phosphate group, right, that is hydrophilic, and it's got two fatty acid tails that is hydrophobic. And so what you have is you have the structure of this compound that's got a hydrophilic or water-loving part and a hydrophobic or water-hating part. And when you look at the structure of the phospholipid bilayer, what you can see here is that the hydrophobic tails of both layers are actually facing each other. And then what you have is on the outside, on the external areas of the lipid, of the cell membrane, you've got the hydrophilic heads over here in the round red balls, right? And so what this means is that it separates out the water that's outside the cell from the water that's inside the cell. Because what happens is that it prevents free movement of substances across the lipid bilayer. This property of phospholipids, where you have a hydrophilic N and a hydrophobic N, makes it amphipathic, right? And the word amphipathic means that it's got a charged hydrophilic N and an uncharged neutral N. And as I said, the arrangements of the phospholipid in a bilayer allow separation from the water outside the cell, the extracellular fluid, from the water inside the cell, the intracellular fluid. So you've got the explanation that's over here of the phospholipid bilayer. Moving along, when we look at the cell membrane as a whole, what we see is that you can see that it's made largely of phospholipids here. The bilayer is over here. But what you can see is that there are also lots of different things that are attached that go through the phospholipid bilayer section. And so a couple of the things that are found within the phospholipid bilayer are lipids. So you've got cholesterol and you've got glycolipids. So remember, glyco means that there's a carb component, and lipid means that there's a fat component. So glycolipids have got a fat and a lipid component to it. We also have a few different types of proteins. So one type of protein are glycoproteins. So you see the glycoproteins over here. And so glyco, again, there's part carb and part protein. And what you can see here is glycoproteins always protrude on the extracellular side of the cell membrane, because glycoproteins are cell identity markers to distinguish self cells from non-self cells. So some of you may know what blood type you are. So you can be either type O positive or O negative. You can be type A, B positive, or type A, type B blood. And those types actually is an indication of the type of glycoproteins that are attached to the outside of the red blood cells. So again, it distinguishes your cells from other cells, right? So, you know, in the world of COVID that we live in, it's spike proteins are glycoproteins. We also have integral membrane proteins. So integral membrane proteins, you've got it over here, are proteins that insert itself along the entire thickness of the cell membrane. And integral membrane proteins include things like channel protein that we're going to talk about later on in this slide. It includes transporter proteins, and it includes a whole host of other proteins as well. So integral membrane proteins span the entire width of the membrane. And thirdly, we've got peripheral membrane proteins. So peripheral membrane proteins don't span the entire width of the membrane, so they're only found jutting out inside the cell, or some of them jut out outside the cell, but they don't span the entire width of the protein. So an example of a peripheral membrane proteins are enzymes that are found on intestinal cells, and they're only found on one side of the cell membrane wall. So that's the brief overview of the cell membrane. Next, we're going to talk about fluid environments around the body. Again, we've touched on this briefly in a previous lecture. So basically, the fluid environment around the cell can be described very succinctly as either fluid that's found inside the cell, and so that's referred to as being intracellular fluid. So intracellular fluid is the fluid environment inside the cell, whereas extracellular fluid is the environment that's outside the cell. And interstitial fluid is an example of an ECF that's found outside of blood vessels. All right, and you can see here interstitial fluid make up a large component of extracellular fluid. And what you can see here is interstitial fluid is the fluid that's found surrounding the cell. but it's not the fluid that is inside the blood vessels. As I said previously, what happens is substances and water inside the cell are kept separately from the substances and water outside the cell by the plasma membrane. The plasma membrane is what we call selectively permeable or semi-permeable. And what semi-permeable membrane means is that it allows certain substances but not everything through it, right? So in terms of the selective permeability of the plasma membrane, it allows water to either slip between the phospholipid tails because water is a small enough molecules, or there are protein channels called aquaporins. And if you look at the name, aqua refers to water, porins refers to pores. So aquaporins are an example of that integral membrane protein that I talked about in the previous slide. And the purpose of aquaporins, if they're found in the cell, is to allow free movement of water across the cell membrane. Some small nonpolar materials like lipids, gases, and alcohol can go through freely across the main portion of the phospholipid bilayer, whereas water-soluble materials like amino acids, glucose, electrolytes, ions are stopped by the main portion of the phospholipid bilayer. And so this begs the question, if you have sodium and potassium inside and outside the cell, we know that a large part of sodium is found outside the cell, and most of the body's potassium is found inside the cell. So how do we ensure that levels of sodium is high outside the cell and levels of potassium is high inside the cell? How do we ensure that potassium and sodium, amongst other things, can actually go in and out of the cell, right? And the answer comes in with the fact that, again, the cell membrane is semi-permeable and that there are pores or ion channels that allow these charged particles to cross the cell membrane. And we're going to talk about the ion channels in a little bit. But first, before we go and talk about that, what I'm going to talk about is the idea of a semi-permeable membrane. So again, semi-permeable is the same as selectively permeable because it controls the movement of substances across it. It allows some substances, but not all substances, to move freely across the membrane. And so what we have here is a picture of a beaker that's divided into two compartments by this red dotted line, which is a semi-permeable membrane, right? And what you can see here is at the start of this thing, you can see that the level of water in compartments A and B is the same, but there are less solutes inside A, right? The purple dots compared to the number of solutes inside B, right? So what we can say here is that when we talk about the concentration of solutes, side B has a higher concentration of solutes because it has more solutes than side A. But when we talk about the concentration of water, what I'm going to say is that side A has a higher concentration of water than side B, right? So the semi-permeable membrane would allow either water to go across it or the solutes to go across it. And in the next slide, what I'm going to say is that we'll have the situation where we will allow water to travel across the semi-permeable membrane, but not the solutes, right? So what happens if water can travel across the semi-permeable membrane, but not the solutes? And in order to actually talk about that, what I'm going to do is introduce the concept of tonicity and of a solution. So the concept of tonicity refers to the concentration of solutes in a solution. So When you have two compartments like a beaker and you're comparing side A and side B, what we're going to say is that in an isotonic solution, what happens is A and B will be isotonic to each other if they contain the same number of solutes. So each of those sites, say, contains 5 solutes, and so that means it's isotonic. If we're talking about a hypotonic solution, what happens is that one site contains less solutes than the other. So in this particular situation, site A will be hypotonic to side B, right? Hypertonic means that there's a higher concentration of solutes as another solution. So when we compare side A and B, we will say that side B has got more solutes in it. So side B is hypertonic compared to side A. And so what you've got over here, if you break down the words, you can actually figure out what they mean. Right? So if you break down the words, each of these terms have got the word tonic, have got the suffix tonic in it, right? So as we say, tonicity refers to the concentration of solution. Now, if you look at the prefix, the first part of the word, iso means the same. So isotonic means that the concentrations of two solutions are the same with each other. Hypo means less than. So in a hypotonic solution, one has a lower concentration of solutes than the other. And then in the third term, hyper means more than. So a hypertonic solution has got a larger number or a greater or higher concentration of solutes as another solution. So let's put this to the test. We've got two beakers here, right? We've got beaker 1 and beaker 2. So in beaker 1, water, but not solutes, can move across the semi-permeable membrane. So in beaker 1, the solution, if we look at the solutions inside A and B, the solution inside A is hypotonic to the solution inside B, and the solution inside B is hypertonic to the solution inside A. If water is the only thing that can move, the movement of water from a higher concentration of water to another, what happens is osmosis will occur, and what happens is water will move from side A to side B. So what happens is water will move that way, not the solutes. So the purple dots don't move. It's the water that will move from side A to side B. In beaker B, water, again, but not solutes, can move across the semi-permeable membrane. And what we can see here is if you count the relative number of dots and compare it with the volume of water in side A to side B, what we can see is that there's roughly the same concentration of solutes in side A compared to side B. So side A is isotonic to the solution in side B, and the solution in side B is also isotonic to the solution in side A. And so what happens is osmosis, the free movement of water, will not occur because there's no imbalance to address, right? Because there's the concentration of solutes in side A is the same as side B, so there's no net or overall movement of water across the membrane. So actually, if you really wanted to know, this is what happens when you have in the situation. So this would be the start of the experiment where you put more solutes in side B compared with side A. And then if you leave it over time, what happens is water will move from side A here to side B to give you this isotonic solution at the end of the experiment. This type of movement of water actually happens in and out of red blood cells. So this occurs when we place a red blood cell into a beaker that has a solution with a different tonicity to the tonicity of the red blood cell, right? So remember, you've got the red blood cell. The red blood cell has a specific tonicity, right? And we're again assuming that the cell membrane of the red blood cell is semi-permeable, which allows free movement of water in and out. So if you look at the example in the first figure, what you have is and if you look at the direction of the arrow, what you can see is the amount of water moving into the cell is the same as the amount of water moving outside the cell. So there's no net movement of water across the cell membrane of the red blood cell. And so what we can say here is that in this example, in this scenario over here in 1, the red blood cells were placed in an isotonic solution because there's no net movement across, and the effect of this is nothing. So the red blood cells stay nice and healthy, maintain their shape, maintain their function. In the second example, what you can see here is that there's a lot of water that moves out of the red blood cells into the surrounding solution. So again, the water will move from a region of high concentration to low concentration, and I'm going to guess that the red blood cells were placed in a hypertonic solution, and so we want to dilute the hypertonic solution that the red blood cells are in by moving water out of the red blood cells into the beaker. The effect of this is that the red blood cells, as you can see here, shrink or shrivel. And if it goes on too long, it undergoes a process called crenation. And you can see that it loses its shape. And so it can also lose its function. In the third scenario down below, there is more water entering the red blood cells from the beaker, the solution. And so I'm going to say that the solution that the red blood cells are in is hypotonic because there's more water outside in the solution compared to the red blood cells. And so water will go from a high concentration outside the red blood cells into the red blood cells. And what happens is when you have a lot of water moving into the red blood cells, then the red blood cells will swell, right? Will get bigger. So you can see here, the red blood cells are very, very plump compared to a healthy red blood cell. And you can see here in the middle of the picture towards the right that this red blood cell has taken in so much water that it's actually exploded. And so the official word for explosion of red blood cells is lysis, or if you want to get fancy, it's hemolysis, right? So hemo refers to red blood cell, lysis refers to breaking apart, right? So this is what happens when our cells are subjected to, as long as our cells are in an isotonic environment, they're fine. But if it's subjected to a hypertonic environment or a hypotonic environment, it can either shrink or it can swell up, and that's not good either way. So we've talked about water moving across a semi-permeable membrane. But I'm going to move on and talk specifically about movement of substances across semi-permeable membrane. So water moving across a membrane down the concentration gradient does not require any energy because it moves down the concentration energy gradient. And this movement is called passive transport. So in passive transport, energy is not required, but it requires a concentration gradient. And so what happens is that with passive transport, substances move down the concentration gradient from an area of high concentration of the substance to an area of lower concentration of the substance. So when we look at the beaker, if we're wanting to talk about passive transport, if the semi-permeable membrane allows solids to move but not water, then passive transport would have solutes in the beaker move from side B, where it's got a higher concentration of solutes, to side A. But if it's a semi-permeable membrane that only allows water to move, the water will move from side A to side B, right? But and it requires no energy because it's like falling down. It's like gravity. You always fall down, you never fall up, right? Or when you talk about Niagara Falls, you can see the water just falling down, right? So passive transport is what naturally occurs if things are left to their own devices. Active transport, on the other hand, requires energy. And that's because active transport goes against the concentration gradient. So the energy that's required in active transport can take the place of can take the form of either adenosine triphosphate or the energy released by the movement of another substance. And we're going to talk about that in the next few slides. So again, if you look at the same beaker, if we want to if we want to demonstrate what active transport looks like in this beaker, if we want to actively move substances, move the solutes against the concentration gradient, then what we will do is we will move the solutes in the beaker from side A to side B, right? Active transport can also refer to movement of substances across the membrane in bulk. So you have bulk movement of substances, proteins that the cell produces. You've got movement of say bacteria into the cell in order for the bacteria to be destroyed. Those also are considered bulk movement. And again, I'm going to talk about it as we go along after I talk about the different types of passive transport in the next slide. So over here, we've got different types of passive transport. So the overall term to describe a lot of different types of passive transport is diffusion, right? So diffusion here is a movement of substances from an area of higher concentration to an area of lower concentration. Osmosis is the diffusion of water through a semi-permeable membrane. So you can see here, you've got the start and the end. So if this is a semi-permeable membrane in the same beaker that only allows water to move across and not the solutes, what happens is this is the start of the experiment. Water will freely move across from side A to side B, and this is the end. So at the end of the experiment, what happens is you have the same concentration of solutes in both sides of the beaker. The term simple diffusion is depicted in the diagram down below, And in simple diffusion, what happens is you don't need any help for the substance to move across the semi-permeable membrane. So usually the only substances that can move freely across the semi-permeable membrane are molecules that are small and don't have a charge. They are lipophilic, they are hydrophobic, they are not positively charged or negatively charged. So again, if you look at this series, there's 3 compartments to it, and this is taken over time. So this is the start of the experiment, and this is the end. So you can see at the start, there's a lot of small uncharged molecules on one side of the membrane, and then as time progresses, more and more will go across the membrane to the other side, and then at the end of the experiment, there's an equal amount of solutes on both sides of the membrane. Sometimes what happens is you've got larger substances that want to diffuse across the membrane, but they can't do it either because it's too large, right? Or it's the substance has got a negative charge or a positive charge. So if the solute is too large, for example, glucose, what happens is glucose can diffuse into the cell with the help of this integral membrane protein called a carrier protein. And the carrier protein just allows glucose, this oval green substance here, glucose to actually bind to the carrier protein on one end, on the outside of the cell. And the carrier protein will actually just transport the glucose through the cell membrane, and then it comes out, pushes it out on the other side, right? Without the carrier protein, glucose can't enter into the cell. If the solute has got a charge, so like the small green circle, what happens is it also requires an integral membrane protein, and in this occasion, the integral membrane protein is called a channel protein or a pore, and again, that allows the small charge ion to actually cross the cell membrane, right? So we've got pores that allow different ions to go in, and other pores that allow different ions to go out, to diffuse out, right? But again, facilitated diffusion always goes from a region of higher concentration to a region of lower concentration. And then the third type of passive movement is called filtration, where you have movement of fluid and solutes from a higher hydrostatic pressure to an area of a lower hydrostatic pressure. So this occurs in the kidneys. In the first function of the kidney is filtration, where you have things, water and solutes moving from an area of high pressure, water pressure, to an area of lower water pressure. All right, so these are the 5 types of passive transport. We're going to go next and look at active transport. So, as I said, active transport can mean transportation against their concentration gradient. So, when we talk about transportation of substances against a concentration gradient, it requires energy. So the energy can be either adenosine triphosphate or the energy that's released when one substance diffuses across the membrane, right? So let's talk about the first one that uses ATP. So the active transport that uses ATP is called primary active transport. And an example of a primary active transporter is the sodium potassium ATPase pump. So as you know, cell membrane, outside the cell, inside the cell, you can see here that the concentration of sodium outside the cell is very high, whereas the concentration of potassium inside the cell is very high, right? So sodium is found largely outside the cell, and we need to keep it this way. So what happens is the sodium potassium ATPase pump actually pumps sodium outside the cell, right? Using ATP as an energy source. So ATP gets loses one of its phosphate group to become adenosine diphosphate with only two phosphate group. And when ATP is converted into ADP, what happens is it pushes 3 sodium ions out of the cell, and it pulls in 2 potassium ions into the cell. And the sodium potassium ATPase pump actually ensures that potassium levels remain high inside the cells and sodium levels remain high outside the cell, right? So it maintains an electrical gradient across the membrane. We're going to talk about electrical gradients much later on. The other type of active transport is the secondary active transport. So we've got the two types of secondary active transport. And in each of these types, what happens is the diffusion of sodium from a region of higher concentration outside the cell to a region of lower concentration inside the cell actually releases energy. And that energy is then used to transport a second substance across the cell membrane. So the example in the middle here is an example of a SIM porter. So SIM refers to same, right? Porter refers to transport. So a SIM porter transport a second substance, in this case glucose, in the same direction as the transport of the first substance, right? So sodium enters into the cell. And so what happens is the sodium glucose symporter then uses the energy that's released by sodium moving into the cell to transport glucose into the cell. The other type of secondary active transport is an anti-porter. Again, anti means against. Porter refers to transporter. So sodium hydrogen ion antiporter uses the energy released when sodium enters, diffuses into the cell to actually push protons or hydrogen ions out of the cell. And see, the direction of hydrogen transport outside the cell is opposite to the direction of sodium transport into the cell. I'm not going to talk about electrical gradients in great detail, apart from noting that most cells have got an electrical gradient, so a difference in the electrical charge when you compare the inside of the cell compared to the outside of the cell. So when you look at electrical gradient, nerve cells have got a negative gradient inside the cell relative to its extracellular environment. And again, we're going to talk about this later when we talk about neurons and nerve cells. The other type of active transport is bulk movement of substances. So substances can move in bulk into the cell or can move in bulk outside the cell. Usually when you transport substances in bulk, it uses packages called vesicles to actually move things across the membrane. And so this is a very efficient way of moving it. So vesicles are like bags. What do we use bags for when we go grocery shopping? We use bags to put in more than one article of groceries so that we can actually carry a whole host of different things, grocery items. Sorry, that we buy from the store into the house at the same time, right? So instead of carrying, one pack of eggs and one thing of milk, we combine them together and we can transport them in at the same time. So in the same way, vesicles are used in the same way. So Movement of substances into the cells is called endocytosis, right? Endo means inside, cyto refers to cell. So endocytosis is moving inside the cell, and moving outside the cell is exocytosis, right? Exo refers to outside the cell, right? So you can see here there are several different kinds of bulk movement into the cell, depending on what things are being moved into the cells. So if there is large movements of large particles, e.g. bacteria, into the cells, it's called phagocytosis, right? So you've got these large particles over here. They, what happens is when they enter in, it actually pinches off a part of the cell membrane to create this vesicle or vacuole. Sometimes water needs to move into the cell. And so what you can see here is you've got this water molecules over here. And so if we want to move water into the cell, then again, a part of the cell membrane is pinched off. And then you've got a vesicle that contains water inside it. So it moves water together with whatever's dissolved in the water into the cell. The third type of endocytosis is specific transport of substances, and that's done through these receptors. So you see these receptors, they bind to the substances called ligands. And so what happens is that the receptor binds to a specific ligand that fits the receptor. And then what happens is it pinches off a part of the cell membrane to actually create a vesicle. And you can see here in the vesicle here, you've got a bunch of receptors with ligands attached to it. So it brings in things like iron into the cell as well as other things. When we move transport substances in bulk out of the cell, it's called exocytosis. And so exocytosis occurs when the cell produces materials such as hormones and neurotransmitters or other proteins, and it wants to export it out of the cell. And so what happens is those substances gets packaged together in vesicles, and then what you can see is the vesicles then move to the cell membrane and then fuses with the cell membrane, and then you can see that it opens, and then whatever substances is released into the extracellular fluid. So we've got movement across the cell membrane, passively or actively. I'm going to talk a little bit about the cytoplasm, the second part of the cell, the cytoplasm, as well as the organelles, right? So you've got the definition of this. So cytoplasm is the internal environment of the cell, which consists of the cytosol and organelles. Cytosol is a jelly-like substance that uses water as a medium, and organelles are basically baby organs that function inside the cell. And like organs in our body, each of these different organelles have a unique function, right? Some organelles are kept inside lipid bilayer membranes. So for example, here, the mitochondria up the top, it's got a lipid bilayer around it in order to actually keep it contained. Another one would be lysosomes again, has got a lipid bilayer around it. Peroxisome have got a lipid bilayer around it. And then we also have the nucleus, which is a type of organelle, and the nucleus contains the DNA of the cell, which then controls the function of the cell. I've grouped the different organelles into their functional category. So the first functional category of the organelles are those that are involved in synthesis, right, in creating things. So we've got the endoplasmic reticulum. So They're highlighted. There's two different types of endoplasmic reticulum. There is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And the reason why they're called rough and smooth is that if you look in the second picture down in the middle there, the rough endoplasmic reticulum have got these bumps that are actually ribosomes, whereas the smooth endoplasmic reticulum doesn't have the bumps, so it looks a lot smoother, right? So in terms of the rough endoplasmic reticulum, as I said, it contains ribosomes, and ribosomes are used to actually produce polypeptides, which then become functional proteins, right? Rough ER synthesizes proteins, smooth ER synthesizes lipids and carbs, stores calcium, and also detoxifies certain substances. Another organelle that's involved in synthesis is called the Golgi apparatus. So if you look at the location of the Golgi apparatus in relation to the endoplasmic reticulum, what you can see is that the rough ER is right outside the nucleus, and the Golgi apparatus is a little distance away. And what you can see is that it's got two sides. It's got a cis side and a trans side. And what happens is the product of the rough endoplasmic reticulum actually travel to the cis side of the Golgi apparatus, and then the Golgi apparatus then synthesizes, modifies, packages the product of the rough ER, the polypeptides, packages it up into vesicles so that it can be transported in bulk outside the cell. The next slide goes through the process of protein synthesis. So to start off with, what happens is it starts off in the nucleus with the components of the nucleus producing a set of instructions for protein synthesis. Then what happened is those instructions is transported out of the nucleus into the rough ER, and that sends a signal to the ribosomes over here to actually assemble the polypeptide. The polypeptide is then folded into its different shapes in the rough ER and placed into these transporter vesicles, right? Because transporter vesicles are like bags, help to bulk transport material. And so these transport vesicles help to transport proteins, which are these funny shaped things, from the rough ER to the cis phase of the Golgi apparatus. And then in the Golgi apparatus, it gets processed and packaged, right? And then released at the trans site. So this is the trans phase over here. So it gets released and repackaged again into secretory vesicles. And so what happens is that the secretory vesicles then transport the packaged proteins into the plasma membrane, where they can be released to either be inserted into plasma membrane or to be exported to be used outside of the cell, right? So this is a mechanism to actually to produce proteins, to transport them, and to release them to where they should go. We're going to talk about this later in a little bit more detail in the next lecture. Other organelles that are found typically inside a typical cell are listed over here. So mitochondria are surrounded by a lipid bilayer, and so that's the structure or the anatomy of the mitochondria. And you can see here it's got these folds or cristae in the mitochondria that help to increase the surface area inside the mitochondria. And the purpose of the mitochondria is to produce ATP, adenosine triphosphate, which is used as a form of cellular energy. The second organelle is a lysosome. Again, the lysosome is surrounded by a lipid bilayer, and inside the lysosome there is a bunch of enzymes, and the function of these enzymes inside the lysosomes is to break down and digest foreign material or unneeded and damaged cellular particles. So you need that cell membrane in order to actually contain the damaging properties of the lysosomal enzymes, right? So lysosomes are like, I guess, garbage disposals or even knives. It breaks things down. So you want it in a controlled and protected environment. And so you've got the bulk transport of bacteria into the lysosomes, and then the enzymes actually digesting the bacteria so that it can be removed and not affect the person. Third kind of enzyme is peroxisome. Again, lipid bilayer to actually contain the enzymes that are found inside it. And the enzymes in the peroxisome are involved in lipid metabolism and also to detoxify certain chemicals. And then the fourth organelle is a proteasome. So you can see here a proteasome is a hollow cylinder. So what happens is proteins go in one end and out the other end, you've got amino acids, right? So the proteins get broken down into its component amino acids. And so the proteasomes are used to break down abnormal or misfolded proteins or proteins that are no longer needed. So as it goes through, it gets broken down into its components. Separate slide on the nucleus. So the nucleus is the largest, most prominent organelle in most cells. Some cells, like muscle cells, have got more than one nuclei, whereas mature red blood cells have no nuclei at all because the nucleus gets ejected as the red blood cell matures. Function of the nucleus is to control the function of the entire cell. Contains DNA, which provide genetic instructions used to build and maintain an organism. Nucleus has got a lipid bilayer surrounding it, and through it you've got this, so the lipid bilayer is called the nuclear envelope, and you can see that throughout the nuclear envelope you've got these holes or these channel proteins that are called nuclear pores, and they allow proteins, RNA, solutes to actually go in and out of the nucleus. The stuff inside the nucleus, the watery substance inside the nucleus, is called the neoplasm, right? Similar to the cytoplasm in the cell. And then the nucleolus inside the nucleus is the one that manufactures RNA, ribonucleic acid, that is necessary to construct ribosomes. Moving along, I'm going to end this lecture with talking about cytoskeleton. So again, if you look at the word cytoskeleton, cyto refers to cell and skeleton is the framework. So the cell is organized, right? There is organization inside the cell, and the organization inside the cell helps keeps things in its own place, right? So the cytoskeleton are like the walls in a building. Instead of just having a huge tent or a huge cavern or a huge garage, most houses and most dwellings and most buildings are separated out into different compartments. And what happens is the cytoskeleton helps you to do that. The cytoskeleton does that with the help of fibers. So there are 3 different types of fibers: small, medium, and large, right? So the smallest fiber is called micro filaments, contains actin chains, and it helps to create a furrow during cell division that we'll talk about in the next slide. The actin also is involved in muscle contraction. Second, middle thickness of fiber is called the intermediate filament. contains keratin chains and has another function altogether. The third type of fiber is the thickest one. So it's the microtubules. And so you can see here, you've got microfilament, intermediate filament, and microtubules. And so these are the ones that help to position organelles inside the cell, helps to maintain the cell shape, and so on. And then I'm going to finish off with how some of these, in some cells, we have these extensions of the cytoskeleton. So some cells have got cilia, and you see the cilia over here. And so they consist of microtubules, and they help to move dust, mucus, bacteria towards the mouth and the nose. So they're found in the respiratory tract, or the cilia are also found in the fallopian tube in the female reproductive system to help to move eggs that are released by the ovary towards the uterus. The second type of cytoskeleton is called microvilli, and microvilli are found in epithelial cells in the small intestine. So this is a microvilli right here. So you can see here they're very, very small, right? And what happens is they increase the surface area so that the small intestine can do its job. And the job of the small intestine is final digestion as well as nutrient absorption. So if you increase the surface area, you can have a lot of nutrient absorption occurring at the same time. Third type of cell extension is the flagellum or flagella, and that is found at one end of the sperm cell. So you see this is the flagellum, and so you can see that it's a tail. And it consists of microtubules, and the flagellum, when it is moving, helps to actually move the sperm cell towards the female egg cell, right? And then the last cell extension is the centriole, which is found in the nucleus of a dividing cell. And again, what happens is the centriole then helps with the separation of DNA during cell division. And again, we're going to talk about centrioles, flagellum, microvilli, and cilia when we actually get to that part of the course, when we're talking about cell division, when we're talking about reproduction, when we're talking about absorption, when we're talking about respiration. And on that note, I'm going to finish up. Thank you.